Welcome back all. We'll continue in this cell signaling course from where we left in the last class, where we were discussing how cytokinin signaling mediated by two component signaling has its own effect on shoot development, root development, vascular differentiation, meristem maintenance, as well as the aspects of cytokinin signaling through generation of type A response regulator and how it crosstalks with the photomorphogenic signals. So, if we say what exactly is the photomorphogenesis, and if we can summarize what we saw in the previous class. So, this was the histidine kinase responding to cytokinin. This generated the AHPs. These were the hybrid type, so it has histidine which was getting phosphorylated, and it has the aspartate residue where this phosphotransfer was happening. Now, these were affecting the expression of type B response regulator, which was feeding back onto type A response regulator, and this type A response regulator was working through the phytochrome B interconversion. This is active, this is inactive. So, this was far red light and this was red light. The presence of type A was ensuring that this conversion is blocked. The active form going to inactive in the presence of far red, or blocking this conversion and then giving more time the phytochrome B to induce the signaling. So, what exactly phytochrome B signaling, when it is activated, what it does? So, basically it responds to the light, and you can understand that light falls on all upper parts of the plant. Which has shoot, leaf, meristem and other structures like petiole, node and so on. So, when photomorphogenic response sets in, it helps in germination. That is something which is happening beneath the ground, but whatever signaling starts, it culminates into the germination of the seed. It is responsible for the inhibition of stem elongation. When germination happens, generally you will have less of a stem elongation, and these leaves from the cotyledon, they come out and they grow bigger in size. So, that stem elongation process early on is inhibited, so that leaf elongation and other things can happen, and that is mediated by the phytochrome signaling. So, longer the phytochrome remains in active form, these processes will be mediated. Also, you need more and more leaves to have these chlorophyll. So, whether it is chlorophyll synthesis or the chloroplast development in the mature form, so that it can do the photosynthesis, phytochrome B is mediating that. Similarly, if you think about the leaf expansion, so that this will be coupled with more and more chloroplast being enriched over there, whether it is shoot branching, whether it is flowering or senescence, all will be mediated by the phytochrome signaling. Remember that some activity are directly regulated by virtue of phytochrome being active, and some responses would be there because phytochrome must go into inactive form also. and we can understand that an active form will be formed by the presence of far red light. So basically in dark period where there will be no light, the photomorphogenic responses will not be there, and the process in which the dark light is mediating certain responses is called scotomorphogenesis. So basically phytochrome will be playing important role in ensuring an equilibrium between photomorphogenic responses and scotomorphogenic responses. We will see more into the other signaling which are affected by the two component system. We saw how cytokinin signaling was affected, how cytokinin signaling was adding to the phytochrome signaling, and we know that the histidine kinases of hybrid type or variant type in plants, they do help in the process of ethylene signaling. So let's see what happens in case of ethylene signaling. I really need not tell that what is the main function of ethylene. We know that it is for the fruit ripening. But it has own set of development related responses, and for any of these responses which is part of the two component system, you will definitely need different components, and one of the prime component here is the receptor, the ethylene receptors. There are two subfamilies of ethylene receptors. There will be hybrid type, there will be canonical types, there will be normal type, and there are variant type. So one of them is called ETR1, ethylene responsing. This is hybrid type. Then you have ERS1, ethylene response sensitive, that's a canonical type. So you have histidine kinase receptor of this type, right? Now what you can understand from ETR1, that ETR1 will have domain arrangement like this, where there will be a histidine and there will be aspartate. This will be the domain which will sense the signal, and here the signal is ethylene. I also discussed in previous class, one of the previous classes, that ethylene being gaseous molecule, it travels inside the cell and it finds its receptor over there. Normally these receptors are bound to the ER membrane, and the binding of ethylene happens to those domains which are bound to the ER membrane. There are other type of receptors which is present in the other subfamily of ethylene receptors, something called EIN4, which is ethylene insensitive and ethylene response. They are hybrid and variant type. What exactly it means? That hybrid, they will be like this, and variant in the sense that histidine is mutated. And then you have ERS2, which is canonical type and variant type, which means that you will have only the histidine domain prominently. There is additional domain but not receiver type, because this is your receiver type domain. And when you say variant, here again it could be something like this. Histidine is mutated to as glutamate or some other residue which is incapable of accepting the phosphate group. So if we see different types of ethylene receptors, you find all possible combination: hybrid type, canonical type, hybrid and variant, canonical and variant. Together they will be serving to mediate the ethylene signaling process. You will find some of these molecules membrane bound in combination with one of the receptor. You will find some of these molecules as a mediator of the signaling. Some we will see that there are response regulators which are also present over there. So how does the signaling happen in case of ethylene? As I said, this is a receptor which is present at the ER membrane. This is your cytoplasmic domain. Here, this is the region which is in the part of the membrane. You will find ethylene binding to happen here. Normally what happens that your ethylene, when not present, this molecule binds to another thing which is called CTR1. It's basically copper transporter kind of thing which binds to this ER membrane and activates it. So what it does, there's another molecule which is ethylene insensitive which works with the ETR, this is ethylene receptor. Combination of these two ensure that the system is receiving the message that ethylene is absent. And how does that happen? That here EIN2 is phosphorylated and stays in the vicinity. Now when ethylene is present, so this is your ethylene molecule. When it binds to the ETR1, it brings a conformational change. Because of that, what happens that this EIN2 is released. EIN2 cannot be phosphorylated and as well as it is released from this vicinity. Now this released EIN can move inside the nucleus. So you can easily assume that this molecule should have an NLS, nuclear localization signal, or it will need something which can take it inside the nucleus. So you saw how presence of ethylene changed the conformation of the receptor which affected the phosphorylation status of EIN2. And the location of EIN2 is sensitive to its phosphorylation status. It is either in the ER membrane proximity or it is going into the nucleus. Now normally what happens in absence of EIN2 inside the nucleus, normally when ethylene is not there, EIN2 is not inside the nucleus. In that scenario, this particular set of proteins, EBF12, they actually inhibit the factors which help ethylene response factor to transcribe. And this is happening with this basically EIN3 and EIL1 complex is degraded by ubiquitination. So in absence of ethylene, EBF12 is inhibiting or degrading EIN3 EIL1 complex, which was sending the ERF1 a signal for transcription. So in absence of ethylene, there is no signal for transcription. But when you have ethylene present, EIN2 moves into the nucleus, it inhibits the EBF. You will notice in multiple examples we will see that how inhibiting an inhibitor is a signal for response to move forward. Yesterday we saw that in previous classes we saw that how kinase was kept inactive by binding of an inhibitor, and you need an anti-inhibitor to activate the system. So you can think a similar scenario which is happening here, that ERF1 which is kept inactive by this inhibitor or something which is degrading the activation molecule. Now this process is activated by inhibiting the inhibitor. And this ERF will activate the transcription of response genes. Remember earlier I said that many of the times these signaling will culminate into the nucleus, where a transcription factor will help in the transcription of set of genes which will mediate the final response. Now this is how your ethylene signaling is working through the ETR and EIN kind of receptor-like molecules. Now, what happens in case of a response regulator? There are some things which come into play, which is as a pseudo response regulator. We will see example of how pseudo response regulators, they help in plants to maintain a diurnal cycle. We all understand that diurnal cycle, everybody has a sleep wake cycle, and this is directly coupled with the light which is being received. So similarly, plants also have a diurnal cycle which probably starts with the morning, with the sunrise, and ends with the sunset. So we understand that these pseudo response regulators play a very important role in this diurnal cycle. We saw in previous cases, whether it was ethylene signaling or many other cases, that variant form of receptor histidine kinase was working. Here we are taking example of variant of response regulator, which is called pseudo response regulator, and how do they add into the diurnal cycle of plant. So canonically, there are response regulator of two types. You can say that this is A type, which has the basal minimum receiver domain with the conserved aspartate residue, or you have a B type, which has in addition a domain which gives it additional functionality. But in both cases, your aspartate residue, where final phosphate group will be received, is intact. The pseudo response regulators, they have the mutation here, and instead of D as aspartate residue as a receiver, you have the glutamate residue. Now this molecule will not be able to receive the phosphate group. Phosphate group receiving is inhibited, because of which it will not help the canonical process. It will need some other receptor or some other response regulator to mediate the process. So we saw multiple different types of response regulator, a master regulator, and so on. Such pseudo response regulator, they not only diverge the signaling but also sometimes serve as a competitor to inhibit the signaling or block the signaling when it is not required. Now, if you think about different types of response regulator, as we saw in case of Arabidopsis, it is known that this domain, which is the receiver domain, there is a specific set amino acid which is known to be present at a particular residue, 2 aspartate residue and a lysine residue. In the mutant ones, this is the one which receives the phosphate group. This has been changed into glutamate. Often it has been seen that the other aspartate residue is mutated to the asparagine. Thus, it will affect the response regulator function. There are multiple genes which are coding for pseudo response regulator present in the plant. So you will notice that in all those, if you align the sequence of these pseudo response regulator, you will find that in place of this canonical aspartate residue, you have this glutamate residue. Now, what basically it will end up doing is that the response regulator is not activated. So in some sense, the catalytic activity or the transcription factor activity, which was part of this molecule, is compromised. If we see how these different response regulators are expressed and how they respond, so if I just simply show you the relation, so authentic response regulators, if I say ARR1, ARR2, they are in one side of the branch, and then other side has PRR 1, 3, 5, 7, 9. 5 of them, a PRR quintet is known. So what you notice that they have diverged at some point of time, and although they are related to the ARR, authentic response regulator, but they form a different group. Now in this scenario, if you look at the expression of these, how do they affect the diurnal cycle? So what has been noticed is that if you consider the light and dark period, dark, light, dark, light, this is how a plant is given the light. It undergoes certain hours of dark period and then certain hour of light period, and notice that how the expression of these molecules changes. So what you would notice that there is a kind of relation of different PRRs coming and going. So they do express in a way, in a fashion, that one of the PRR is high and the other PRR is low. So then how it relates with the signaling process, there we can see that in this scenario, which if you notice, we are talking about the quintet of PRR. So this is, if I say PRR 9, 7, 5, 3 and 1. And this is a gene which is important for hypocotyle development. This is called late elongated hypocotyle. Hypocotyle is the region below the cotyledon which should grow only to the size before it should grow only to a definite size before leaves are out and expanded and mediate the function. So you have late elongated hypocotyle gene which is here, and what you notice here is that if you compare a 12-hour window of light and dark cycle, this is 6 AM in the morning, this is 6 PM in the morning. So you notice how the expression of this late elongated hypocotyle gene is correlated with the light. So you can consider this also to be a gene which may be coupled with the photomorphogenic response, right? Because this is coming up. So CCA is coming up, and with respect to that, you see how different PRRs are coming. Gradually over this 12-hour window, one of the PRR comes and it goes down, second PRR comes and it goes down, third PRR comes and it goes down. What we still do not completely understand is that these molecules are coming up, they are being synthesized, they are pseudo response regulator. So how do they contribute to this diurnal cycle? Remember that since these are variant type of response regulator or pseudo response regulator, they might compete with the actual response regulator. They might inhibit some of the processes which the authentic response regulator was doing. One of this response regulator, which is PRR1, or if I say PRR1, this is also called true oscillator component. Why? You can clearly see that this particular gene, which was the late elongated hypocotyle, that came up early in the morning when the light responses started. Now you notice this particular true oscillator component molecule, which is the pseudo response regulator 1. When is that coming up? That is coming towards the end of the day. Right? So the oscillation with respect to the early morning molecule, which is late elongated hypocotyle, and this true oscillator component, it's like they are in sync, that when first molecule comes, other molecule is not there. So there seems to be again something which we have been telling, that there is a feedback regulation. So when this late elongated hypocotyl molecule comes up in the morning, it ensures that PRR is not produced. And when PRR is made, it gives the feedback that okay, at this point of time the CCA or LHY should be expressed. So a continuous feedback between the two molecules as true oscillator component, one coming up early, one coming up late, there is very little overlap. That's what will be giving this diurnal cycle a particular form. What we still don't understand is that what goes on by these, what we still do not understand completely, that what is wrong, what is mediated by the pseudo response regulator in terms of regulating the diurnal cycle. So what we have understood today is that plants use the two component system for photomorphogenic signal, where type A response regulator ensure that phi B remains in its active form for long, and we also realize that this type A response regulator were being generated from the cytokinin signaling. Then we saw how nicely ethylene signaling is regulated by the hybrid and the variant two component system, where ETR1 and EIN2 played great synergistic role to ensure that the ERF1 activity is regulated when ethylene is present. And then we saw that how pseudo response regulators, which are variant in its receiver domain, 5 of them, quintet, their expression is correlated with the day time, the period, the amount of light and so on, and one of the PRR, which is PRR1, which also looked like a true oscillator component in combination with CCA or long hypocotyl gene. So we notice that two component system plays very important role in many different types of signaling mediated either by cytokinin or by ethylene.